Hello, today I'll provide an update on the ICH M12 guideline, which addresses drug interaction studies. As you'll hear during the presentation, the expert working group is finalizing edits based on comments received during the public consultation period. I will start with a background that includes general principles related to evaluation of drug interactions, the objectives and scope of the guideline, and the timeline for the expert working group from start to finish. The table of contents displays the full scope of topics included in the guideline. Due to the number of topics included in the guideline, I will focus on 5 topics that generated discussion at various points during the working group's deliberations. The topics are in vitro cutoff values, drugs with high protein binding, endogenous biomarkers, studies with concomitant medications, and the interpretation of study results. General principles that support the development of the guideline are as follows. Drug interactions can occur when patients take more than one drug, which may impact safety or efficacy, resulting in altered risk-benefit. The evaluation of drug interaction potential should be risk-based and proceed in a stepwise manner from in vitro to clinical, and it often includes predictive modeling. The evaluation should begin early in drug development to avoid unnecessary exclusions in clinical trials and to ensure the safety of clinical trial participants. The timing and utility of non-clinical studies, clinical studies, and predictive modeling is dependent on clinical context and the type of product. A common consideration is the intended patient population and other drugs they need to take. The interpretation and translation of drug interaction study results should be based on an understanding of the variability of the drug exposures and the exposure-response relationships for desirable and undesirable drug effects. The objectives of this guideline are to develop recommendations that promote a consistent approach in designing, conducting, and interpreting in vitro and clinical drug interaction studies during development of a therapeutic product, and to reduce uncertainty for the pharmaceutical industry to meet the expectations of multiple regulatory agencies, which may lead to more efficient use of resources. There's a wide range of potential topics related to the scope of drug interaction evaluations during drug development. The scope of this guideline includes pharmacokinetic interactions with a focus on enzyme and transporter-mediated interactions, and it focuses on small molecules and biologic products, and the specific biologic products are monoclonal antibodies and antibody-drug conjugates. The guideline does not address pharmacodynamic interactions, pharmacokinetic interactions due to gastric pH change, formation of complexes or chelates, or food effect, and it does not address interactions with new modalities such as oligonucleotides. The working group selected the scope based on topics in the greatest need of global harmonization and for which adequate scientific information is available to provide detailed instructions. The drug interaction topic was proposed in November 2018. The initial work started in June 2019 with the formation of the expert working group at the 2019 meeting in Singapore. Due to the pandemic and travel restrictions, the working group conducted all meetings virtually in 2020 through 2022. We collaborated effectively in spite of the virtual setting and the time difference. The draft guideline was published in May 2022, with public consultation period ending that November. 2023 was an active year with comments discussed when we met in Geneva and finalization of the guideline revision soon after our meeting in Prague. We are starting the various clearance steps, and we anticipate adoption of the guideline in March. In addition to the guideline, we are working on a question and answer document and educational materials to support the implementation. During the public comment period that closed in November of 2022, 30 stakeholders provided approximately 1,000 comments, and the comments were distributed across the various sections. The introduction to the guideline includes the objective, background, scope, and general principles that support the details outlined in the guideline. The in vitro evaluation section describes the process for evaluating metabolism-mediated interactions, transporter-mediated interactions, DDI potential of metabolites in vitro. This section includes cutoff criteria for determining whether the in vitro study results translate to a need for clinical evaluation of drug interactions. The clinical evaluation section describes various types of clinical studies and considerations when planning the studies. I want to point out that we include information about an emerging topic, endogenous biomarkers, and I will describe that topic later in my presentation. The guideline provides information about pharmacogenetic variations in drug metabolizing enzymes and drug transporters to the extent that they affect evaluation of drug interactions. And there is a brief section that describes drug interaction considerations for therapeutic proteins. There is a section that describes reporting and interpretation of clinical drug interaction study results, which directly informs risk assessment and management. The detailed appendices support the studies and analyses described in the main body of the guideline. The glossary is a new appendix that defines terms used in the guideline. Another new appendix provides details about the evaluation of protein binding because the accuracy of the methods is essential for interpretation of in vitro results. Other appendices describe in vitro methodology for metabolism and transporter studies, predictive modeling, and it provides a list of drugs that can be used in vitro and clinical studies. Finally, we provide a list of references that support emerging areas and key topics. However, it is not an exhaustive list of references. With that overview of the contents of the guideline, I will use the rest of the presentation to focus on 5 topics: in vitro cutoff values, considerations for drugs that are highly bound to plasma proteins, endogenous biomarkers, clinical studies with likely concomitant medications, and the interpretation of study results. Cutoff values compare an in vitro measure of inhibition or induction with an estimated clinical exposure to determine whether a clinical drug interaction study is recommended. The cutoff values are of high interest to drug developers because they directly affect the number of clinical drug interaction studies needed. The working group considered the following factors when selecting cutoff values: consistency among regional guidelines, in vitro in vivo analyses based on literature and FDA and EMA approved products, the impact of capping protein binding estimates for drugs that are highly protein bound, and the likelihood of false negative predictions. The next topic is the handling of drugs with high protein binding. The draft guideline indicated the following regarding protein binding measurements. There are uncertainties in the accuracy of protein binding measurements for highly bound drugs, those with greater than 99% protein binding. Due to uncertainties, the fraction unbound in plasma should be capped at 1%. However, it also indicates that there have been advances in the methodology and it's an active area of research. Thus, in some situations, measured fractions less than 1% can be used if the accuracy and the precision of the measurement is demonstrated, and this includes the provision of validation data, including bioanalytical method and appropriate positive controls. Based on comments received during the public consultation, the working group discussed the need for additional clarity regarding expectations related to bioanalytical methods, when positive controls are needed, and whether multiple protein binding experimental approaches are needed. As we revised the guideline, we invited external experts from industry to present protein binding evaluation results. We also conducted in-depth literature review on the topic. Following this work, we revised the guideline for clarity and added an appendix that describes considerations for protein binding measurements for highly bound drugs. Endogenous biomarkers is another emerging area that the guideline addresses. The draft guideline indicated the following regarding the use of endogenous biomarkers. Recent literature describes the potential utility of endogenous substrates for some drug transporters. Evaluation of change in exposure of the endogenous substrate in the presence of investigational drug may provide information about the drug's potential as a transporter inhibitor. Based on comments received and continued advancements in this emerging area, the working group discussed the need for additional information in the guideline. Similar to the work for protein binding, we invited external experts from industry to present results from their evaluation of this topic, and we reviewed recent literature. In the end, we revised the guideline to clarify how this emerging area can contribute to drug interaction evaluation. During the consultation period, we received a range of comments requesting more clarity on clinical studies with likely concomitant medications. As a reminder, the draft guideline indicated that drugs with a well-understood and predictable pharmacokinetic and drug interaction properties regarding level of inhibition, induction, or metabolic pathway are known as index drugs. Studies with likely concomitant medications often follow index studies. The specific concomitant drugs for evaluation are selected based on mechanistic understanding of the potential for drug interactions and the relative frequency of co-administration. The studies are often informative to patients and medical professionals, but the results may be difficult to extrapolate to other drugs. Currently, we have identified index drugs for CYP enzymes, but not transporters or UGT enzymes. Thus, drug interaction studies for these pathways often include concomitant medications. Based on comments requesting more clarity on specific studies with concomitant medications, the working group further discussed the topic. In general, the selection of potential concomitant medications for drug interaction studies is case by case, and it depends on the therapeutic area, the intended population, and the safety and efficacy properties of the drugs. We consulted with an external expert to refine the list of UGT substrates and inhibitors included in the guideline appendix, and we conducted literature and database research to refine the list of transporter and UGT substrates and inhibitors included in the guideline appendix. The last topic I will discuss today is the interpretation of study results. While writing the draft guideline and addressing comments, this was a topic of high interest. The guiding principle is an emphasis on the use of exposure-response information to determine no-effect boundaries for the drug as an object. An object is a drug that is affected by an inhibitor or inducer. The term victim has been used previously. No-effect boundaries represent the interval within which a change in systemic exposure measure is considered not significant enough to warrant clinical action. The point estimate of the ratio, so with or without the inhibitor or inducer, is normally evaluated in relation to the no-effect boundary, but variability should also be taken into consideration. In the end, we consider all available evidence when interpreting the results. I will close my presentation by acknowledging the numerous individuals who worked on the guideline over the past 5 years. We also appreciate the time that external groups and individuals spent providing valuable comments on the draft guideline and the work of subject matter experts at various regulatory agencies and companies. We are looking forward to the implementation of the guideline. Thank you for your attention, and I look forward to questions during the question and answer period.